For centuries, humanity believed genes were fixed, permanent, unchanging, locked into place inside chromosomes forever. Scientists believed inheritance followed strict rules. Genes passed from parent to child, always staying exactly where they belonged. It sounded logical. But one woman would eventually discover something so shocking, it would completely change our understanding of life itself. Her name was Barbara McClintock, and for years, almost nobody believed her. The story began in the 1920s. Genetics was still a young science. Researchers across the world were racing to understand heredity, chromosomes, and the hidden instructions inside living cells. Most scientists believed genes were stable and predictable. But McClintock noticed something strange. While studying corn plants under a microscope, she saw genetic patterns behaving unpredictably.
Some corn kernels suddenly changed color. Spots appeared where they should not exist. Traits switched on, then disappeared. At first, the results seemed impossible, because according to everything science believed at the time, genes were supposed to stay fixed forever. But McClintock kept seeing evidence suggesting something unthinkable. What if genes could actually move? The idea sounded absurd. Genes moving inside chromosomes? Most scientists rejected the possibility immediately. But McClintock trusted the evidence she saw under the microscope. Day after day, year after year, she continued studying the mysterious behavior inside corn cells. And slowly, a revolutionary truth began to emerge. Certain pieces of genetic material appeared capable of moving from one location to another inside the genome itself. And when those genes moved, they could change how other genes behaved. Some genes switched off. Others switched on unexpectedly. The genome was not fixed and motionless. It was dynamic, alive with change. Then, in the 1940s and early 1950s, Barbara McClintock introduced one of the most important discoveries in modern biology: transposable elements, later known as jumping genes. Tiny sections of DNA capable of moving throughout the genome. Today, the idea sounds fundamental, but at the time, many scientists simply refused to believe it. The scientific world dismissed her work. Some researchers ignored her entirely. Others believed her conclusions were impossible. And because technology at the time was still limited, McClintock could not fully prove the molecular mechanism behind her discovery. So for years, her research remained largely overlooked.
But she never stopped believing the evidence. Then, decades later, science finally caught up with her discovery. By the 1960s and 1970s, researchers began discovering transposable elements not only in corn, but in bacteria, plants, animals, even humans. Suddenly, Barbara McClintock's once impossible theory became undeniable scientific reality, and the discovery transformed genetics forever. Scientists realized jumping genes played major roles in evolution, mutation, genetic diversity, immune systems, and even cancer research. What once sounded impossible became one of the foundations of modern biology.
Then, in 1983, more than 30 years after her breakthrough, Barbara McClintock received the Nobel Prize in Physiology or Medicine. Not for inventing a machine, not for creating a surgery, but for revealing that life itself was far more dynamic and unpredictable than humanity ever imagined. And perhaps the most extraordinary part of the story is that Barbara McClintock discovered the hidden movement of genes decades before the world possessed the technology to fully understand what she had already seen.